All right, so first thing we're going to do is I'm going to go back over some things that we learned last week, just kind of hit the highlights real quick before I move on to new information. Remember last week we started talking about heredity, or how genes and traits get passed down from parents to offspring. We talked about Gregor Mendel, the Austrian monk, who back in the 1800s figured out how things get passed down through his work with pea plants. And he's kind of the father of heredity, or this concept of passing things down from parents to children, such as hair color, eye color, blood type, things of that nature. We talked about the difference between characteristics and traits. Characteristics were really broad categories. As you can see the chart down below, broad categories that he studied in his pea plant would be like plant height, pea color, flower color, things of that nature. Characteristics in the human population would be broad categories like hair color, eye color, blood type. Traits are specific examples of those characteristics. So if the characteristic was hair color, a trait would be black, brown, blonde, red, gray hair. If the characteristic was blood type, the traits would be like AB, O negative, A positive. There's different types of the characteristic. So traits are more narrowed down. We talked about how most plants have both male and female parts. That way they can pollinate themselves. That's called self-pollination. The pea plants were able to do that. If a plant pollinates itself, it creates what we call true breeding plants down here. True breeding plants are kind of like pure-blooded or purebred. They are pure for their characteristics. They don't have any cross-contamination. So if they pollinate themselves, they would create essentially clones of themselves. However, a lot of people don't like that. They want to do this. They want to do cross-pollination, where they take the pollen from one flower or one plant and force it to pollinate the others. And the way they do that is they rip off these pollen centers here called the anthers. When you detassel corn, you rip off the tassel, which is the part that has the male pollen on it. So it can't pollinate itself. It has to rely on something, another plant, to do that. So that's going to cross-pollinate. Cross-pollination is good because it creates hybrids. Hybrids are blends between two parents with different characteristics and traits. Well, not different characteristics, but different traits for those characteristics. So it creates different plants with different qualities, or a blend of those qualities, which you are a blend of your parents. You are not exactly a clone of your mom if you're a girl, and you're not exactly a clone of your dad if you're a boy. You are a blend of the two. So you might have heard your grandparents go, "Oh, you've got your mother's eyes," or "You've got your father's height." So that's kind of where that comes from, because you are a hybrid. You are a blend of your parents. Moving on, we talked about how Mendel, how he got these results. When he did his experiments, he noticed that there was always a trait that seemed to pop up far more frequently than the other trait, and he called that trait that showed up more frequently the dominant trait. He showed the trait that showed up less frequently the recessive trait. He called it the dominant trait because it was powerful and it overpowered or blocked or hid the recessive trait. So that's where the word dominant comes from. Recessive traits only showed up if no dominant genes were out there. So, for example, we used purple cards last week, and we had certain combinations that gave us the dominant characteristic, and we had certain combinations that gave us the recessive characteristic. And let me just kind of pause here, and I'll pop these up on the screen so we can talk about those real quick. So when we did our experiment last week and we modeled one of his examples, when he did the top one up here, he did, if I get my marker out, if he did flower color. So if you pollinated each other. We did that experiment where you had two cards and you ran around the room. If we were early at school, and you would trade cards with other kids, and then you would get certain outcomes. So some of you would have this outcome when you were done. You'd have two purple cards. That means you have two dominant genes, and you would absolutely be the purple color flower. Some of you had this combination, if you would have done that in class, where you'd had one purple card and one white card. The purple card is the dominant gene, the white card is the recessive gene, and the purple dominant gene would mask or hide that recessive gene, and you would still be purple in color. Or you could have this combination. You have two white cards. That means that you have two recessive genes, and you would not have any dominant genes, and you would show the recessive trait, which would be the white flower. As you can see, there's two combinations to give you purple flowers, and there's one combo to give you white flowers. So 75% of the time, the purple flower is going to show up, and only 25% of the time will the white flower color show up. So this is the ratio that he expected, and you can see he got that 75-25 or percent wise, or a 3-to-1 ratio. It's the same thing. You had 3/4 at 75 cents, 1/4 is 25 cents, so 3-to-1 is 75-25. So you can see 3-to-1, 3-to-1. It's not perfect because it's only a prediction of what's going to happen. It might not actually happen that way, but it would be really, really weird if it didn't. It'd be like, okay, what are the odds of rolling a 6 on a dice? Well, it's a 6-sided object, so you got a 1/6 chance. So could I roll a 6 16 times in a row? It would be astronomically rare, but it could happen. So these are all predicted outcomes, the 75-25 or the 3-to-1, and he got pretty darn close every time. When we do this experiment in class, we get pretty close to 75-25. All right, so that's dominant and recessive. Just kind of going back over that really quick, and then we move on. Let's skip this. All right, next part we need to talk about is genotype and phenotype real quick, just to review those. Remember, genotype is the letter combos that you get, and they use letters to represent the genes because it just makes it easy. Please don't think that inside your body you have little genes that have letters on them. They don't. They chose letters because it's easy to use and keep track of. They use capital letters to represent dominant genes, and they use lowercase letters to represent recessive genes. And you just don't want to use a letter that looks the same as a capital as it does a lowercase. So you would not want to use like W because it looks too similar. You wouldn't want to use C because it looks too similar. Because sometimes you guys don't write the correct size. So you want to use a letter that looks different, like, I don't know, B and little b look different. So there's no confusion there. Let's see, what else could you use? You could use D and little d. It doesn't really matter as long as you use lowercase letters that look different than their capital letter form. So just to review here real quick, if I can get this to work. So there's it's actually 3 different kinds of genotypes here. We got homozygous, and there's 2 ways to be homozygous. Homo means same. So homozygous, you could be, and I'm going to use the letter B. It doesn't matter what letter you use. You could be big B, and that would be like having those 2 purple cards. So this is called homozygous dominant. And you could be big B, no, screwed that up. Sorry, erase that. Already did that one. You could be little b, and that's called homozygous recessive. So big B, homozygous dominant. That would be 2 purple cards in that experiment. Little b, homozygous recessive. That would be like having 2 white cards. In fact, this is the, darn it, come on, pen. This is the only way only combination that's going to give you the recessive trait. So there's more than one way to be dominant, but there's only one way to show the recessive. Heterozygous, hetero means different, so that'd be like having big B, little b. That would be the one purple card, one white card, and you would still show the dominant trait because this gene would overpower and dominate that recessive gene. And so there's two ways to be dominant and only one way to be recessive. Those are the genotypes. Genotypes are letter combinations, all right? Then we move on to phenotype down here. Phenotype is what do you look like because of the letter combinations you received. So many students every year get these confused. Genotype is what kind of genes do you have? What do you have? Dominant, recessive? What's your letter combo? That's genotype, type of genes. Phenotype is what do you physically look like? And the way I remember it is phenotype and physical go together, okay? PH, they both start with PH. So that's how I remember. But kids get those scrambled up all the time. And I'll ask them, what's the genotype? And they'll say pink. And that's not a genotype. Pink is a physical description. That's a phenotype. Or I'll do it the other way. I'll say, what's the phenotype? And they'll say, big B, little b. I'm like, no, that's the genotype. So you got to keep those straight in your head. So genotype, letter combination. Phenotype, what do you physically look like because of the genes that you have? All right. Another example here is something called incomplete dominance. Incomplete dominance is a different twist on what we've been doing. All right. All we've been talking about so far is something called complete dominance, where one gene completely overpowers another one. Incomplete dominance is a little bit different. It's a little bit trickier. And here's how it goes. I'm going to pull up some pictures here. So in an incomplete dominance example, before we were just doing complete dominance, where one gene completely overpowered another gene. So for example, if you look over here, you've got two red cards, so that would give you like red flower color. One red card and one white card. The red would dominate the white. You'd get red flower color, and the only thing that would give you white flower color is the one that doesn't have any dominant genes. However, if you're doing incomplete dominance, what happens in the heterozygous form here is that the red color does not completely dominate the white color. It incompletely dominates it. So that means that they will actually blend together. And when they blend together, they would get this combination. You'd get the pink color because these two colors would blend together, and you would get down here, you would get your pink color as such. So that's what's trying to be illustrated right here. Incomplete dominance problems, if you were, I used R, big R, you'd be red flower. If you were little r, the recessive, you'd be the white. But if you're the heterozygous form, you take on a blended color and you'd be pink. You don't know if they're giving you a homework question and they want you to predict the outcome of this genetic cross. You wouldn't know to do incomplete dominance unless they told you in the problem. I don't think I'm going to give you any incomplete dominance problems here at the beginning because I don't want to confuse you. I'm just going to do straight up 2 colors. I'm not going to do 3 colors. I'm just going to show you what incomplete dominance is, but I don't think I'm going to actually physically do any on a worksheet because I just don't think you're ready for that. All right, so if you have another example here of something called codominance, co means like together, like you could be co-workers and you work together. You could be like co-champions, you tied for first place and you both share the trophy. So that would be another example of co, so 2 at the same time. So in codominance, you could have this combination here, and that would give you red flowers. You could have this combination here with two recessive genes and you'd have white flowers. But in the codominant form, if you had the blend and you had one dominant gene and one recessive gene, you would turn out with having both colors at the same time. So co means together. So you'd have red and white at the same time and you'd be stripy or splotchy. For example, I use this bunny rabbit here. If he was like Big B, he would be black fur and he'd be completely black furred rabbit all the way through. If he was little, two little B's, he would have white fur. But if he had one big B and one little b, he'd be heterozygous. And if he was codominant, he'd be splotchy, and he is splotchy. He has splotches of black and splotches of white. So that's what codominance is. Both colors or both characteristic traits show up at the same time. All right, so just to kind of review here, let's use the bunny. So you've got three different kinds of ways you can be. You can be completely dominant, complete dominance, incomplete, and you could be co. All right, so let's use the bunny. So if we were doing complete dominance, if he was big B, he'd be black. If he was little b, he'd be a white bunny. If he was big little, heterozygous, if you're doing complete dominance and you're heterozygous, that means the black color would completely overpower the white color and he'd be still a black bunny. There is no third combo when you do complete dominance. And when I give you your Punnett square worksheet today, that's all I'm going to do. I'm just going to only do complete dominance problems. I'm not going to have a third color option because I think it will mess with your mind. Incomplete dominance would be a blend. So in the bunny rabbit, if he was big B, he'd be black. If he was little b, he'd be white. And if he was big B and heterozygous, he would be a blended color because that's what it means to be incomplete. No one gene completely dominates the other one. So they blend, and guess what color he would be if he was incompletely dominant? That's right, he would be a blend of black and white, which would be gray. So he'd be a gray rabbit. If he was codominant is, if it's big B, he would be black bunny. If he's little b, he'd be a white bunny. And if he was big B, the heterozygous, he would be co. That means both equally show up, and he'd be the splotchy guy right here. So that is how that works. All right, last thing on the list here, Punnett squares. Punnett squares were developed by a man named Reginald Punnett, and they are pretty simple, but you have to kind of get the hang of them. And Punnett squares, this is the little box over here that he came up with. It looks like a window. It's got four quadrants in it. And it is a device used to predict the outcomes of genetic crisscrosses. So here's how they work. So remember, they're just a prediction. They don't actually say what actually is going to happen. It just helps you predict. So what you do is you are given your scenario of individuals that you're going to crisscross. So in this case, they are telling you that the dad's genetic combination for this trait is big B, and the mom is also big B. And you put them into your Punnett square. You put one parent at the top, and you put one letter above one column of boxes, and the other, got my cat is up here visiting me. Please go away, cat, I'm trying to work. All right, so you got dad's up top, big B, mom's on the side, and you could have switched those out. It doesn't matter which parent's on the top part, which one's on the side here. And you do a crisscross. It's kind of like playing Battleship where you say like A7, you crisscross them up. So for example, this would be the big B comes down, this big B goes over, so this one's big B. This box here would be big B and little b. This box here would be the crisscross of this big B and this little b. And this box here would be the crisscross of these two, that one coming together, and this one coming together. So you just crisscross them up, and you look at your combos. So if you look at your combos, they will ask you, if I could, come on, eraser, why aren't you erasing? There we go. Erase, 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 erase. They will usually ask you on Punnett squares to list what genotypes are possible and list what phenotypes are possible and their percentages. All right, so here we go. Genotype. Remember, that is your letter combination. What letters can you get? Well, in this crisscross, I see big B as a possible outcome, and I see big B, little b as a possible outcome, and I see little as a possible outcome. I see all three outcomes are possible with this cross. What are my odds? What's my ratio? Well, one of these boxes gives me big B, little b. In fact, I'm going to erase that so you can see better. Come on, eraser, for Pete's sake, erase. Erase. Why aren't you erasing? You're erasing terribly. There, that's better. Erase, 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 erase. Come on, 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 come on. All right, so one box gives you big B, big B. So that's 1 out of 4 boxes, so that's 25% chance of being that genotype. I see 2 boxes of this combination, so that's 2 out of 4 boxes. So when I went to school, that would be 50%. And this last dude here, little b is 1 out of 4, so it's 25%. When you do these problems, if these 3 numbers don't add up to 100, you done messed up, because it has to add to 100% of the people. All right, those are your genotype combinations. All right, now in the problem, it'll tell you what your trait is. So I don't know, let's say B stands for blood type, or no, I don't know, let's make it whether you can roll your tongue or can't roll your tongue. Let's do that. All right, let's say dominant is you can't, or you can do it. Say dominant is you can do it. All right, so this combination is dominant, so that's going to give you the dominant phenotype of I can roll my tongue. This is going to be the dominant phenotype. So it'll ask you to predict the phenotypes now. So I'm going to write phenotype. Phenotype is what you actually are going to look like. And there are 2 choices. So one choice is I can roll my tongue. I'm just making this up. And the other one is I can't do it. I cannot do it. All right, so can roll, let's say that's dominant. There's 2 ways of being that. You could be this combo and you can roll your tongue. You could be this combo and you can roll your tongue. So add these 2 together. 50 plus 25 is 75%. So you got a 75% chance of rolling your tongue. And those that can't, 25% chance. All right. However, if you are a parent, a mom and dad here, is it possible to have 4 kids that can't roll their tongue? Absolutely. Each individual kid has a 25% chance of not being able to roll their tongue. But there's a chance every single time. It's rare that you would have 4 kids that all can't do it, because this is the most likely outcome. So that's what Punnett squares do. They predict the most likely outcome. I'm going to do a couple examples with you, and then I'm going to give you a couple questions that you'll do for homework. Let's get a new slide here. Okay, so the last part of this video is going to be showing you how to do Punnett squares and answer questions based upon them. And at any time, if you think, okay, I got it, I understand Punnett squares, I'm ready to move on to the worksheet, you can stop the video and move on. I'm going to do 4 problems, and if you get the hang of it after 1 or 2 problems, then more power to you. You don't need to watch the rest, but you can always come back and watch them later. All right, so here we go. Let's go to work here. So example problem number 1. It says, in rabbits, black fur is dominant over white fur. Show the cross of a heterozygous black bunny male with a homozygous white bunny female. Use the letter B to represent your allele color. All right. So what we want to do here is work our way through this. and they want us to use the letter B. And my homework questions, if they don't tell you what letter to use, pick whatever letter you want as long as it looks different as a lowercase than it does a uppercase letter, okay? All right, so this one it says, come on, what is the genotype of Daddy Rabbit? All right, so Daddy Rabbit says he is a heterozygous black bunny. Okay, so he is heterozygous, so he's big B, little b. It's got to be. And what is his phenotype? All right, remember, we're only doing the ones that are completely dominant. We're not doing codominant, and we're not doing... okay, I'm looking at my neighbor who's out in the yard doing something weird. What is he doing? Okay, whatever. Okay, so anyway, we're not doing the blended ones, and we're not doing the codominant or incomplete dominant. Just flat up, 2 color combos, 2 choices, that's it. All right, so Papa Rabbit here, he's got 1 dominant gene, so he's got to have the dominant fur color, and then the problem, it says that black is the dominant fur color, so that is his phenotype, what he physically looks like. Next, what is the genotype of Mommy? Mommy is a homozygous white bunny. Okay, so she's white. That is the recessive quality, so she's little. And what is she going to look like, her phenotype? Well, in the question it says she was a white bunny, so she's got white fur. And the next question is, what are the possible genotypes of the baby bunnies? Okay, well, we don't know that unless we crisscross the parents up. So let's do it. So we put our letter combos on here. So this is dad and this is mom. And it doesn't matter which parent we put on the top part and which one we put on the side. So it doesn't matter, get the same outcome. So I'm going to put dad on the top part. Big B, little b, that's dad right there. And mom down here was little. All right, so we crisscross these up. So this box would be a cross of the big B on top and the little b on the side here, so it'd be big little. This box here would be this B and that B crossing, so little, because I'm just crisscrossing the boxes up. I'm bringing that one over here. Okay, this would be a cross of these 2 dudes, so that'd be big little. And this last one would be this guy and this guy crossing, so it'd be little. So that is the possible outcomes here. So possible genotypes of the baby bunnies. We'll switch colors here. Let's see, I see this possible outcome. So big B, little b. Remember, genotypes are the letters. And I see this possible outcome. Little. Those are the only 2 outcomes I see possible letter-wise. That's it. Then it says, what are the possible way they're going to look, phenotypes? Well, if you're big B, little b, you're going to have the black fur. And if you're little, you're going to have the white fur. All right, so they can have both black and white colored baby bunnies. What are the odds of having a black baby bunny rabbit compared to a white baby bunny rabbit? All right, well, let's see. What are the odds of this versus that? Well, let's switch color. Let me erase. Switch colors here, actually. These 2 boxes have a dominant gene in it, so those 2 boxes are going to be the black bunny rabbit, and these 2 boxes will be the white bunny rabbit. So 2 out of 4 boxes for each one. So 2 out of 4 is 50%. So that's a 50% chance of being the black bunny. And there's going to be a 50% chance of being the white bunny rabbit. All right. And is it possible for 5 baby rabbits to be born of all of them having black fur? Yes, it's absolutely possible. Each time it's a 50-50 chance, as we just showed here on G. Just like having a boy versus having a girl, you could have, each time you have a kid, it's a 50-50 chance of being a boy or girl, but could you have 5 girls in a row? Sure, You beat the odds, but it could happen. So, is it possible for 5 baby rabbits to all have black fur? Yes. And it says to explain, and my explanation would be Punnett squares only predict the odds, not what actually might happen. Let's go ahead and take a look at another problem here. This one says, tall is dominant over short in pea plants. Use the cross of a homozygous short plant, which is plant number 1. And it's going to be crossed with a homozygous tall plant, which is plant number 2. Use the letter big T to represent your genes or alleles, same thing. All right, so plant number 1 says it is homozygous short, so that is going to be little t, little t. What is the phenotype of that dude? Well, he's little t, so he's going to be short. Plant number 2. Plant number 2 is homozygous tall. So he is going to be big T, big T. And what's he going to look like? He's going to be tall. I don't know why it took me so long. It's going to be tall. All right, what are the possible genotypes of the new plants? All right, well, we got to do a crisscross here. So we need to take this plant and cross them up. Doesn't matter where you put them. I'm going to put one plant up here, big, big. I'm going to put the other plant here, little, little. I'm going to crisscross it up. Big T, little t for that box. This box is these two combinations, big T, little. I'm going to have these two crisscross here, that's big, little. I'm going to have these two crisscross here, that's big, little. Well, everybody's big, little. What are the possible genotypes? That's the letter combos, and I only see one combo here. Is the only thing that kids can be is big, little. What are they going to look like? Well, if they have one dominant gene, and we're doing complete dominance, which is all that I'm going to do, they're going to have the dominant characteristic, and a dominant characteristic was going to be tall. So they're going to be tall. Okay. What are the odds of having a tall plant? Well, every single box up here has a combo that has a dominant gene in it. So everybody's going to be big T, little t, and everybody's going to be tall. And I don't know why I'm writing the word tall, because that's not what they asked. They want to know the odds. The odds are going to be 100% tall. Is it possible for the 2-parent plants to have a kid that's short? No. Because there is no outcome of that is little t, little t. So impossible, not going to happen. Okay, the next one we're doing here is about ears. So ears, like, sometimes they're attached to the bottom part where like your earring would go. Sometimes they're really loopy, and you can kind of flip the bottom of your ear. Sometimes they attach straight to the side of your head, and you don't have much of a loop. So that's what they mean by free earlobes or attached. So it says that free earlobes are going to be the dominant here. And we have 2 parents are both heterozygous. They want us to use the letter E. So both parents are heterozygous. So the genotype of the parents, heterozygous is one of each. That's big E, little e. What is the phenotype? Well, we're going to have the dominant characteristics since they have a dominant gene. And the dominant is up here. It says free ear. So they have free ears, dangly. What are the possible outcomes of the kids? All right, well, we got to cross these 2 parents. They're both big, little, big, little. Crisscross them up. This one would be big, big. So these 2 crossed. These 2 crossed would be big, little. These 2 crossed would be also big, little. And the last box would be little, little, because it's those 2 crossed. All right. What are the possible genotypes of the kiddos here? Well, I see one kiddo is big, big, so I'm going to throw that down. I see big little as a possibility. I'll throw that one down. And I see also little as a possibility. So all three combos are possible. What are the possible phenotypes? What are they going to look like? Well, if they have at least one dominant letter, they're going to be the free ear people, or dangly. Well, that's got a dominant letter, and that one's got a dominant letter, these two. So, and this guy's going to give the attached ear. So both possibilities are going to happen here. So we can have free kids, and we can have the ones with the attached ears. We can have them both. What are the odds of having a kid with attached ears? Well, to be attached ears, you got to have two littles, and that's only one box out of the. Here, let me erase this. Oh, I erased more than I wanted to. How do I undo? Change colors here. Change colors. Yeah, this is the only one that's going to give you the attached little. So that's only one out of the four boxes. So that is a 25% chance. Or you could say 1/4. Either way is fine. Could the parents in the problem have 3 kids, all of who have attached ears? Well, this is the attached ear. And every time they have a kid, they have a 25% chance of having attached ears. But could they have 3 kids in a row? Yep. Each time they have a kid, it's a 25%. They're going to be beating the odds, but it's possible. So it's possible because they can have a little e kid. All right, one more to go. If I can get this to work, there we go. All right, last one here. We have bumpy seeds are recessive to smooth seeds. Show a plant that always produces bumpy seeds. Crossed with heterozygous smooth seeds producing plant number 2. Use the letter B to represent your alleles or genes. All right, parent plant 1. Plant 1 is bumpy. And it says bumpy or recessive. So, and use letter B. So bumpy's recessive, and he's bumpy. So he's going to be little, because that's recessive. And what is his phenotype? Well, it tells you he's bumpy. So he's B-U-M-P-Y, bumpy seeds. And parent number 2, plant 2 was heterozygous. Okay, so heterozygous is big B, little b. And what do they look like? The dominant one, bumpy seeds are recessive to smooth seeds. So smooth is dominant. So he's going to be the dominant characteristic, because he's got a capital letter in his genotype. So he is going to be smooth. What are the possible genotypes of the new plants? Okay, well now we got to do the Punnett square. Why is my neighbor driving back and forth like crazy? Okay, let's see. Big B, little b is one parent, and big B, little b, little b is the other. So we got this parent and that parent. We're going to cross them up here. All right, let me blow this up. So this is big B, little b. This is big B, little b. This is little b, little b, little b, little b. All right, there's my boxes. And answer the questions. What are the possible genotypes for the new plants? Possible genotypes. Okay, well, I see big B, little b up here as a possibility. And I see little as a possibility. Those are the only 2 letter combos. What are they going to look like? Well, if you've got one dominant gene, you're going to be smooth. So these big little guys are smooth. And these little guys are bumpy. What are the odds of having a bumpy seed plant? Well, these 2 boxes give you bumpy. So that's 2 boxes out of 4 boxes. 2 out of 4 is 50%. And that's it.